Welcome back, children. So I think you saw the restriction endonuclease, how it will work on the plasmid vector and also the target gene or foreign DNA. So Escherichia coli. Now look at the slide. Escherichia coli means the all the base pairs and 6 base pairs only, and it is very particular about 6 base pair, and it always used to cut fifth dash G to A and again down opposite again G to A. So it will cut only in that particular area. So that is first enzyme echo R1 Escherichia coli. Like that, you just take a screenshot or write and keep in your note regarding the remaining enzymes. That is not given your NCERT, but for your understanding I have given all those things. For your competitive exam you can use. Now this enzyme, second enzyme, it's a provident castrated. In that organisms the base pair is also 6, and here the location is different. Okay, it is though the see the base pair. Okay, see the base pair. The base pair is different from that of previous one, so that is only we call it as a recognized site. So the echo R1 will recognize only these base pair, and it will work only between GA. So automatically it will cut, will get a sticky end. Okay, the sticky end overhang means that is a sticky end. In that regions we can see the insertion of our foreign DNA. Like that, this organism has a different part of base pair. Even another is a third enzyme. Here also we can see 6 base pair. Here the sequence is different. And fourth one, Haemophilus. Here the base pair is only 4. Okay, here the base pair is only 4. And Haemophilus, another organism, here also base pair is 4. To understand how it will work on specific site, and it will work on between a particular base pair, and how it is identifying palindromic sequence only I gave this slide. Okay, I think now you are thorough with that. So here and all it is blunt end, it is not a sticky end. We can't able to stick anything for the blunt end. So only for sticky end we can able to insert the foreign DNA. This we can use it for other technique. Overhang means we can stick, we can paste, we will get a recombinant. This blunt end we won't get. We can't use it for recombinant DNA. We can use it for other technology. Is it clear, children? So next we'll move on to, this is what I have explained with the help of your book diagram. And now look at the same book, page number 197. This is our foreign DNA. Already what we learned only, we want to see in a pictorial diagram. Foreign DNA. This foreign DNA was cut into small pieces, because DNA means automatically it will has a palindromic sequence at least 3 or 4 places. So all those 3 or 4 places it will cut. Okay, all those 3 or 4 places it will cut. For example, these 1, 2, 3. We got at least 4 or 5 fragments in the one single DNA. Why? Because all those palindromic sequence will be seen in at least 3 or 4 sites. So it will cut all those sites. Even vector DNA has a nucleotide sequence that also will be cutted with the help of the restriction endonuclease. So we are going to select from this. We have so many fragments. So from this we want to select based on size, and which is suitable to this only we are going to paste. So we are doing restriction endonuclease. We are doing cutting of our gene of interest with the help of restriction endonuclease. With that we'll get so many copies. So many copies, every copies won't mix with the DNA. Why? Because the length might be different. Maybe it will cut between or at the corner or at the front. So there will be a length variation will be. So we need to find which length is good. Okay, which length is good. Based on that we'll select the length of DNA, and we can insert into a foreign DNA, and we'll get a recombinant DNA in a vector, that is plasmid, and we are introduced into Escherichia coli. It started to divide, and we'll get a GMO, that is genetically modified organisms, or we'll get a new recombinants in a organisms. So how to select DNA? Okay, how to select DNA for recombinant? For that we have one more procedure is there, that is only we'll call it as gel electrophoresis. That is only we'll call it as gel electrophoresis. Okay, So action of, now look at the slide, action of restriction endonuclease results fragments of DNA. Okay, now fragments of DNA was ready. Now fragments of DNA was ready. So we need to select the DNA fragments. For that we want to use one technique that we can call it as gel electrophoresis. What gel? Agarose gel. What is agarose gel? We are taking from the red algae, a seaweed. Okay, agar. From that we are taking the gel. Okay, that because of that only we can call it as gel electrophoresis. And normally DNA is a negatively charged macromolecule. Why it is negatively charged means, because we know DNA is contains so many nucleotide, sugar, phosphate, nitrogen base, and always this phosphate groups in the DNA backbone carry a negatively charged oxygen molecule, and because of that it will give negatively charged ions to the pentose sugar also. So automatically the whole DNA will become a negatively charged. DNA is negatively charged. Why? Because the phosphate group which is present in the nucleotide is a negatively charged. So this charged ion will be shared even in the pentose sugar. So all those, everything will attain the negatively charged molecule. And okay, this negatively charged molecule DNA is used to select different kinds of fragments based on the negatively charged condition only. So depends on, now look at the slide, depends on negative nature of DNA. External electric field is applied to the medium, DNA moved to anode. Now we'll see the gel electrophoresis. This will be interesting to follow. Now imagine this is all centrifuge tubes. Okay, this is all centrifuge tubes. Okay, these small tubes we can call it as centrifuge tubes. All those centrifuge tubes contains all your DNA fragments. Now all those DNA fragments they will pour in this one. So all this are DNA fragments containing centrifuge. Okay, and here it was poured into the plate. Okay, it is a typical agarose gel electrophoresis. Okay, in that they poured, in that center we can see gel will be, what gel? Agarose gel. So we know DNAs are negatively charged and they will give artificially power supply. If negatively charged, automatically it started to move over the positively charged ions. So it started to move over the positively charged ions. So cathode will started to move towards anode. For that we need to give artificial power supply. So this DNA while moving from cathode negative to positive, automatically agarose gel used to hold DNAs in their different length. Okay, DNAs in their different length. So from that we can, what is our desire? Either we need a very lengthy DNA or we need a very short DNA. Based on that we can select our DNA length. Okay, and here normally it won't give any color. For that we need to stain the DNA. The staining is etherium bromide. Okay, etherium bromide is the staining. How we are using, okay, saffron for chromosomes, like that stain, etherium bromide is used to stain the DNAs, and after staining we need to expose to UV radiation. Afterwards only we'll get this beautiful agarose gel electrophoresis. Will get it used to glitter over the light, and automatically we can able to see the cutting of DNA fragments. So gel electrophoresis is a technique which is very interested to select the length of to select the DNA fragments in their length. For that they are using external power supply. Based on the length, it used to stick on to the agarose gel. They never move because agarose gel means it used to capture it, to hold the DNA molecules. And here what stain is used for DNA? Etherium bromide. After staining, they'll use to expose in UV radiation also. And afterwards, what is our gene of interest? We can select, and that can be, okay, that can be used, and after removing gel, that can be used to make recombinant DNAs. I think now you are clear about the recombinant DNA technology. Now we are clear, and next class onwards we'll see cloning vector. We'll stop with that. So maybe Saturday and Sunday I'll send videos for vectors. Clear, children? So that's all about your gel electrophoresis. Again, I'll go through the points. So medium matrix is agarose gel. So here medium is what? Agarose gel. Now look at the slide. Medium is agarose gel. It is a natural polymer extracted from seaweed. And medium act as a sieve, separate DNA according to their size, smaller fragments further. Separated DNA is stained with ethylium bromide, exposed to UV radiation, and it will bring orange color. All those are very, very important. Ethylium bromide, UV radiation, bright orange color bands of DNA in a gel. Separated bands of DNA cut from the agarose gel and extracted from gel piece. That process we'll call it as elution. Okay, what do you mean by elution? Separation of DNA from the agarose gel, we can call it as elution. Now we'll have a purified DNA fragments. So now this DNA fragment is added to our vector. So our recombinant DNA is ready now. Okay, with that I'll wind up. Next class I'll send videos for cloning vectors. Thank you.